Hello everyone. Now we are going to discuss about the reversible cell injury. When damaging stimulus removed, restoration of the normal cellular function and structure takes place. How this mechanism works? Whenever there is hypoxia, this results into the decreased oxidative phosphorylation and ultimately decreased ATP production. Decreased ATP production will result in decreased functioning of the sodium pump. As a result, there is increased influx of sodium along with water and calcium inside the cell and increase the efflux of potassium. This ultimately results into the swelling of this cell and endoplasmic reticulum and loss of the microvilli blebs occur due to the swelling of the cell. Another mechanism is due to decreased ATP synthesis, there is increase in anaerobic glycolysis, which result into the decreased glycogen level, increased lactic acid synthesis. Lactic acidosis result into the decreased pH or acidic pH and ultimately result into the chromatin clumping. The third mechanism is detachment of endoplasmic reticulum and ribosomes, which result into the decreased protein synthesis. Fatty changes in organs involved in lipid metabolism are also noted in the reversible cell injury, like fatty liver. Now we will see irreversible cell injury. When the injurious stimuli is for prolonged period or severe intensity, this results into the irreversible cell injury, which can be of two types, mainly necrosis and apoptosis. So first we will see necrosis. Necrosis is a pathologic process, essentially pathologic process, that is the consequence of severe injury. What are the characteristics of necrosis? Denaturation of cellular proteins, leakage of cellular content through the damaged cell membrane. These are the cellular organs, their leakage into the surrounding tissue. Denaturation of cellular protein, leakage of the cellular content through the damaged plasma membrane, local inflammation, enzymatic digestion of the lethally injured cell. First is cellular protein denaturation. Second is leakage of these cellular content through the damaged plasma membrane. Third is local inflammation, and then enzymatic digestion of the lethally injured cell. So these are the characteristics of necrosis. There are some entities, DAMPs, damage associated molecular patterns. They are the indicator of severe cell injury, namely ATP and uric acid. ATP is an indicator of mitochondrial damage and uric acid is indicator of DNA damage. Necrosis associated with leakage of intracellular protein through damaged plasma cell membrane. These leaked cellular content then go into the circulation and can be detected by the various tests. For example, in cardiac muscle injury, leakage of contractile protein, troponin, which is an indicator of early indicator of myocardial infarction. In liver cell or hepatocyte injury, leakage of transaminases, SGOT, SGPT. This is one of the commonly performed tests in liver function test to check the liver cell injury. In wild duct epithelium injury, there is release of alkaline phosphatase. Now we will see the morphology of necrosis. Whenever there is a necrosis, there are certain morphological findings, microscopical findings are noted in the cell. Increased eosinophilia due to decreased cytoplasmic RNA or increased accumulation of denatured cytoplasmic protein. The first is increased eosinophilia. Second is glassy homogeneous appearance due to the loss of glycogen. Third is there are myelin figures. They are the precipitates of phospholipid. They degrade into the fatty acid and ultimately result into the calcification. Fourth morphological finding is large amorphous densities in the mitochondria. They are basically calcium deposited along with the denatured protein. Fifth, nuclear features. What are the nuclear features seen in irreversible cell injury? Pycnosis, in which clumping of nuclear chromatin is there. Karyorexis, there is fragmentation of the chromatin material. And karyolysis, dissolution of the nuclear chromatin. Now we will see pattern of necrosis. So we will briefly see the pattern and their typical features. First is coagulative necrosis, and this is the most common type of necrosis in which cellular architecture preserved. This usually occurs in solid organs like liver, kidney and heart. The other type of necrosis is liquefactive necrosis, transformation in which we see transformation of the tissue into viscous fluid. And this is observed in brain or in infection which result into the pus. The third type of necrosis is caseous necrosis. Grossly, we see a cheese-like necrotic material and is seen in granuloma formation and tuberculosis. Fourth type of necrosis is fat necrosis. This is result due to the release of lipases which accelerate the triglyceride ester into the fatty acid, and on saponification they result into the chalky white deposits. Fibroid necrosis is another type of necrosis. This results due to the antigen antibody deposits in arterial wall. These immune complexes and plasma protein then leak out. This is noted in polyarthritis nodosa, malignant hypertension and rheumatic heart disease. Thank you.